yesterday, we talked about the chromosomal theory of inheritance and how it is how chromosome behavior during meiosis um, supports Mendel's theories of independent assortment and segregation. So today we are specifically talking about sex-linked inheritance, or genes that are involved with sex chromosomes. So we're talking about the X and Y chromosomes in regards to humans, and we'll talk about a couple other forms of inheritance. So objective for today, and we're talking about linked genes tomorrow, and then we'll have an activity with linked genes and chromosomal inheritance on Friday. So hopefully by the end of Friday, you should be able to explain what it means for a gene to be sex-linked, how the inheritance pattern works, and provide some examples to support those answers. Before we get started, I want to talk about the difference between sex and gender, just from a scientific context. So, in a biological context, the term sex is the classification into a group with shared anatomical and physiological traits. Traditionally, it's been described as a binary, male, female. But as we learn a little bit more about how chromosomes work and how genetics work, we are coming to understand that there are that should be the word distinct at the very end of that first sentence. But these classifications are a lot less distinct. The likelihood that someone has more or less sex chromosomes than the traditional two is becoming blaringly apparent. So even the concept of biological sex is no longer considered to be a strict binary. So when we talk about sex in a biological term or in a biological context, we'll use male, female, etc. Gender is completely different. Gender is a cultural experience of identification on a gender spectrum, or on a gender binary. So this is where terminology like man, woman, non-binary comes into play. So sex is determined by chromosomes, and gender is more of a cultural influence and personal identification. So this isn't necessarily super critical to the concept of sex-linked inheritance, but I wanted to give a kind of background as to where terminology comes from, and how our scientific experience has kind of expanded upon the traditional idea of what it is, of what biological sex is, and how that is different or similar to the concept of gender. So, I don't think that in any scenario in biology, especially when we're talking about genetics, I will use the term man or woman. If I do, it's just a simple mistake. I will try to stick to male and female. And usually, if you are given a problem or a problem set with terminology, it will also be male and female. So, figured I'd give that little preface. So that's the difference between sex and gender, especially in a biological context. So we are going to talk about the chromosomal basis of sex, how biological sex is determined chromosomally and developmentally, which I think is actually pretty cool. So, I know this side's hefty, but I need to give you the scoop. Biological sex is determined by chromosomes. The existence of some, the non-existence of another. And when it comes to humans, we have X and Y chromosomes. So in mammals, we have two types of sex chromosomes, X chromosomes and Y chromosomes. These next two sentences can be summed up as two X's equals female and XY is male. So an egg will have either an X or well, if goodness gracious, where'd my brain go? So the determination of sex relies on which chromosome the sperm cell brings into fertilization. So if you think about how when an egg is formed, right, the female who is producing that egg only has an X to give. But the sperm cell that is coming into fertilization can either have an X or a Y. So whatever determines that offspring's biological sex is the chromosome that is brought in by the sperm. So it is someone's biological father who determines whether or not they are two X's or XY. So that is how mammals work. So our humans and pets and all that fun stuff. But there are other ways of determining biological sex. Usually, how we determine this is the female is the one that produces the egg cell, and the male is what produces the sperm cell, which is why plants can be considered can have both male and female parts, because part of it produces the sperm cells or pollen cells, and part of them produces the egg cells. So, in humans, the anatomical signs of biological sex begin to emerge when the embryo is 2 months old. So, that is why up until the 2-month scan, someone cannot determine the sex of their child through an anatomical scan, right? They can determine it through a chromosomal blood test, but those are typically tend to be invasive and expensive. So, until you're a human being is about 2 months old, the rudiments of the gonad systems are generic. I should say generic. Maybe I was going too fast when I wrote this. So, if you look at the physiology for the female reproductive system and the male reproductive system, they actually look really, really similar. So the beginning stages of both of those systems look very similar. It isn't until about 2 months in utero that the gene on the Y chromosome, called the SRY gene, is kind of activated. This is why male humans also have nipples, right? They don't need them, but everyone's default female until about 2 months old when this SRY gene kicks in. So the SRY gene is required for the development of testes rather than ovaries. So there's a lot of jokes in there about how if you're a male, then sorry, because the SRY gene. So yeah, no, default is a female genetic system, or not genetics, female physiological system. So if something happens with that SRY gene, then the reproductive system doesn't develop as it could have. So that is how the physiological anatomy of the reproductive system and development happens. So until about 2 months old, everything's generic. After 2 months old, that SRY gene can kick in or not kick in and trigger the development of testes if it's there or ovaries if it's not. So until then, everything's about the same. If you think about how things are shaped, it kind of makes sense as to how they are kind of grown from the same base elements. But there are other systems of sex determination. So in the XY system, so in mammals, we have an XY and an XX. And if an offspring gets two X's or gets the X from the sperm cell, they are biologically female. And if they get a Y chromosome, they are biologically male. We also have the XO system, where some organisms only have one sex chromosome. So they don't have the existence of a Y chromosome, they just have the X chromosome. And if they have both, if they get two copies of this chromosome, they are biologically female. And if they get one copy, they are biologically male. We also have the ZW system. So the ZW system is kind of the flip of the XY system. So in the ZW system, if they have both of the same chromosome, they are male. So if they're ZZ, they are male. But if they have a Z and a W, they are female. And then with the haplodiploid system, if an organism is diploid, they are female. And if an organism is haploid, they are male. So there are a lot of different systems than the one we are familiar with. But in mammalian systems, it's always the XY system. So birds or a lot of birds use the WZ system. And then a lot of insects have the XO or haplodiploid system. There's actually studies going on about how there are technically 3 sexes within a honeybee system, because even though worker bees are genetically identical to the queen, they don't lay eggs. They have the capability of it, but they are never fertilized, so they would be considered a 3rd biological sex. But scientists like to think in binaries because they thought humans were binaries. So we're still kind of breaking these systems down based on reliable scientific information. But these are the 4 basic systems. XY is what mammals have. The XO system, the WZ system, and the haplodiploid system. Any questions here? So, that is how sex determination happens. Now we are going to kind of move into sex-linked traits and X-linked traits. So a gene located on either sex chromosome is called a sex-linked gene. So the SRY gene is a sex-linked gene because it is on a sex chromosome. But the human X chromosome is a lot bigger than the Y chromosome. So there are a lot more genes on that X chromosome. So we have a special name for our X-linked genes. So if a gene is X-linked, it just means that it is on the X chromosome. So things like ocular albinism, Duchenne muscular dystrophy, androgen insensitivity, immunodeficiency, hemophilia, color blindness. We'll all talk about some more specific examples, but those are all considered X-linked genes. But if you look at the difference in size between the X and the Y gene is much, or chromosome, sorry, X chromosome and the Y chromosome. The Y chromosome is much smaller. So we really only have the instructions for how to be a male in there. So Human X chromosome has a lot of different genes on it, and we call those genes X-linked genes. So all X-linked genes are sex-linked genes, but not all sex-linked genes are X-linked genes, if that makes sense. So, inheritance of X-linked genes. I know this is a lot of text, but I don't know where the sentence is going. I know it's a lot of text, but we can do it. So, this first sentence can really just be summed down to, because females get two X's, we have a different pattern of inheritance, right? So the dominant, recessive, codominant, the stuff that we've learned about in autosomes is going to look a little bit different in sex chromosomes. So if a X-linked trait is a recessive allele, right? If a recessive allele is on an X-linked is on an X chromosome, the female will only present the trait if they are homozygous for that allele. So they have to have both of their X chromosomes with that allele on it in order to express that trait. But because males only have one X chromosome, they only have one locus for this trait, the concept of homozygous and heterozygous kind of lose meaning, right? They can't be homozygous if they don't have two copies of the gene. They can't be heterozygous if they don't have two copies for the gene. So we've started using this term hemizygous, which just means they only have one copy of the allele. So if you see hemizygous, odds are they only have one copy of this chromosome. For this reason, a lot more males will have X-linked disorders than females will, because females have a backup. They have two copies of the allele, so if they get the recessive one, they will have a kind of bonus attempt at getting the right, or not the right, but the non-harmful trait. Whereas if someone is born male and they have a recessive trait on their X chromosome, they're kind of stuck. What they got is what they got. So X-linked disorders show up more in males than they do in females. We'll talk about some specifics. I believe what I've got coming up are examples. Everybody understand how in X-linked recessive disorders, why they would show up more in males than they would in females? Okay. So, examples. Red-green color blindness is an X-linked recessive disorder. So this is just an example of how red-green color blindness works. So this is how a person without red-green color blindness would see these apples, and someone with red-green color blindness would see apples. And it is a recessive trait on the X chromosome. And I got a little Punnett square right here. So if this is biological father, and they have a normal allele in their Y chromosome, and the biological mother is a carrier, so she can't she or they sorry they aren't colorblind. But they are a carrier. Um, if so the father can only give the normal allele to his children. So the female offspring of this cross will always be safe. They will never exhibit red-green color blindness because it is a recessive trait, and they're always going to get this dominant allele from their biological father. The biological male offspring for this cross are not so lucky. They have a 50-50 shot. So if they get their X chromosome, if they get the normal allele from their mother, then they don't have red green color blindness. And if they get the recessive allele from the mother, they do. This is actually a really fun example because it's my life. My dad is colorblind. I'm not sure if it's specifically red green colorblind, but he's very bad with colors. When my parents got divorced, he had to start dressing my little brother. It was a disaster. So my father is red green colorblind, which means he has X little b y. Right. So I am not colorblind. My little sister is not colorblind. My little brother is not colorblind. Which means that most likely my mother is homozygous dominant for this trait because there would be a higher likelihood of my little brother being colorblind if she were recessive. So their children, of the three of us that happened, of the male offspring, there is a 0% chance that if I had a brother with my biological parents, that he would be colorblind, right? But this also means that me and my little sister are guaranteed to be heterozygous. So if I chose to procreate and I had a son, then there is a 50-50 chance that he would be colorblind, which is fun to know. But this is like a real life example of how this, but if this were like a more debilitating disorder where a genetic counselor would come into play. So if this were a more serious genetic disorder, apart from just like saying things are the wrong color and getting stuff mixed up every once in a while, if this were a like quality of life altering disorder, a genetic counselor could be brought in to determine, okay, what is the likelihood that I pass this trait on to a child? How would that affect my quality of life and their quality of life? Some specific examples of these more debilitating X-linked disorders. Make sure this is on. Is Duchenne's muscular dystrophy. So Duchenne muscular dystrophy affects 1 in about 3,500 males born in the U.S. And the disease is categorized by a progressive weakening of muscles and loss of coordination. Usually, individuals affected by this rarely live past their 20s. So, researchers have found that the disorder is caused by an absence of a muscle protein called dystrophin, hence the word dystrophy, and have mapped this gene to a specific locus on the X chromosome. So, if this is on the X chromosome, yes, that means a goodness gracious. Males are more likely to have this disorder than females are because females have to get 2 copies of the recessive allele, which is almost impossible because uh, they would have to get a let's kind of squares are so much easier. So if a female were to get Duchenne's muscular dystrophy, right? They would have to get two recessive traits, right? Ooh, that's not how that works. Anyway, in order for this to happen, the male contributor to this person has to have the trait, right? In order for a female to have it, she has to get a copy from their biological mother and their biological father, right? Males with this disease often don't live past their 20s, so it is very rare that someone with Duchenne's muscular dystrophy reproduces. So the likelihood that a female has this disorder is very, very unlikely because oftentimes the males with the disease are unable to reproduce and unable to pass that genetic information on anyway. So usually when someone is born with this genetic disorder, it is because the mother is a carrier. So if we have an unaffected father and a carrier mother, their daughters are 100% safe because they're always going to get the good copy from their father. But the sons have a 50-50 chance. It just depends on which X chromosome their mother gives to them. Does that make sense? Okay. Another example is hemophilia. Hemophilia is an X-linked recessive disorder. I promise not all X-linked disorders are recessive, just most of them, and most of the examples. So hemophilia is an X-linked recessive disorder defined by the absence of proteins required for blood clotting. So people with hemophilia, if they get a cut or a scrape or a bruise, often have to be hospitalized relatively quickly and given artificial platelets in order to stop the bleeding. That way, they do not bleed out. Even something as simple as you fall down and scrape yourself could be disastrous. And in the 1800s, this genetic disorder was widespread among the royal families of Europe, um, which is really interesting because royal families have a tendency to intermarry, especially in the 1800s. So, this pedigree right here shows all of the interactions with hemophilia in the royal family starting back in 1786. So every person in this pedigree with hemophilia can thank Queen Victoria. Because it all traces back to Queen Victoria. So this is an X-linked recessive disorder. This pedigree actually has carriers marked in here. So we know that one of these people up at the top has to have it. They just don't know which one's which, so they can't put that information on here. But she had to get that trait from someone. So she is a carrier. They had 1, 2, 3, 4, 5, 6, 7, 8, 9 children. Of those 9 children, 3 of them got their X chromosome from Queen Victoria. 2 female offspring and 1 male offspring. So, Alice is a carrier. She had 1, 2, 3, 4, 5. No, 1, 2, 3, 4, 5, 6 children. And again, 3 of them got their X chromosome from her, which makes sense because 50-50. Or got the affected X chromosome. So we've got 2 carriers and 1 affected. As you can see, a lot of the times affected males kind of stop the bloodline, except for Leopold right here. He lived long enough to reproduce, but not many, because it was just these two kids right here. Does everybody understand how this trait is tracked through this family? All right, this is a very, very popular image. I would not be surprised if you've seen it before. But hemophilia is an X-linked recessive disorder, and this royal family example is a really good one because A, there's a lot of people in it, and B, we know a lot of information about it, especially considering it is the starts in the 1800s, and down here we've got Prince Harry, Prince William, and all those people. Luckily, they descended from, well, this family that did not get the trait and this family that did not get the trait. As you can see, this arm of the royal family intermarried within this arm of the royal family, and they got really lucky that the hemophilia gene did not come from either end. That's Queen Elizabeth and Prince Philip, that both came from eventually Alice and Edward. Yeah. Any questions so far? All right. Now we're going to talk about X inactivation, which I think is super cool. So, female mammals inherit 2 X chromosomes, right? But we don't make twice as many X-coded proteins. We have double the information, but we don't make double the proteins. And that is because almost all of one X chromosome in each cell in a female becomes inactive during embryonic development. So, someone with 2 X chromosomes, during development, their bodies will just shut off one of their X chromosomes. Doesn't matter which one, in which cell. The inactive X in each cell of a female condenses into a compact object called a Barr body. So, if you have 2 X chromosomes, you got 1 X chromosome from biological father, you got 1 X chromosome from biological mother. Your body is a mosaic, with some of those cells with the maternal X chromosome shut off, and some of those cells with the paternal X chromosome shut off. Doesn't matter which one. So, if you've got 15 cells in your body, like 15 cells in your body, there's a 50-50 shot that each of those cells has condensed the maternal X chromosome or the paternal X chromosome, which I think is super cool. Just wraps it up and calls it a Barr body. In fact, this right here is a nucleus of a cell, and you can actually see the X chromosome all condensed down here, out of the way, kind of pushed off to the side, because they don't need it. It's too much information. They just wrap it up and put it to the side. They don't need it. Which is really smart when you think about it. So, yes. Okay. So, most of the genes in the X chromosome that form the Barr body are not expressed. So, the genes on the Barr body aren't expressed. The only time those Barr bodies are unwrapped is in the ovaries, when those chromosomes are reactivated, so that every egg has an active X chromosome after meiosis. So the only time we unpack that extra X chromosome is when The only time we unpack that extra X chromosome is whenever we need to pass it on to a gamete. And a British geneticist, Mary Leon, demonstrated that the selection of which X chromosome will form the Barr body is completely random. So took a random cell in the body. There is a 50-50 chance that the paternal X chromosome is wrapped up, and there's a 50-50 chance that the maternal X chromosome is wrapped up. This is why we don't make double the proteins that exist on the X chromosome, because our bodies have figured out a way to wrap them up, put them aside, and only use them when we need them, which is during meiosis. That way, an inactive X chromosome isn't passed on to a gamete. It would be really bad if a gamete with an inactive X was fertilized with a Y chromosome, because then that embryo would not be viable if it just had a Y chromosome. We'll talk a lot more about the number of chromosomes given to each offspring when we talk about genetic or chromosomal disorders. So, how do Barr bodies form? The inactivation involves a couple different processes that involve modifications to the DNA itself and the histone proteins that the DNA wraps around. The biggest thing that happens is that the nucleotides are methylated, which means a methyl group, or a CH3, is added to each nucleotide in this chromosome. That way, they literally cannot be read. DNA polymerase, RNA polymerase cannot attach to a methylated nucleotide. Now, when mitosis happens, when mitosis happens, there's a process to undo this, so each cell gets an identical copy. But when a cell is just kind of doing its thing, not dividing, just existing, that X chromosome is all inactive and wrapped up. I think there's an enzyme called methylase that does it, but don't hold me to that. There's a really, really cool example with X-linked inactivation, and that is in calico cats. So, in cats, color or fur color is X-linked. The gene for fur color is on the X chromosome, which means if a cat is more than 3 colors, odds are that cat is a female. So these tortoiseshell cats right here, you can see that some of it is orange and some of it is black, and that it's kind of completely random as to which parts got the orange and which parts got the black. That is because orange and black fur are on the X chromosomes. And wherever the cat has orange fur, it means that the X chromosome with the black allele has been Barr-bodied. And the wherever it has black fur, it means the X chromosome with the orange fur allele got Barr-bodied. I think is super stinking cool. So if you ever find a cat like this that is a male, that is likely because their chromosomes look like that. That is the only way. Is if something happened, something messed up, and a kitty got 2 X's and a Y. which is very, very rare. Not impossible, but very, very rare. So if you ever find one that's a male, that means he has a chromosomal disorder. Super cute kitty cats. Always love a cat example. So I believe that's it. That's all I got for you. There's an exit ticket on Canvas. Go ahead and vote for Snow Coming. If you have the time as well, don't forget to do that. And yeah, if you have any questions about this, let me know. Love to talk about it. Oh, I love it. What's his name? Love it. They're just so great.